So in today's lecture, we are going to look at the concept of ladies and gentlemen, you are welcome. I hope you can all hear me. You are welcome. Please, can you hear me? Hello, can you hear me? Okay, good evening. Let's give some time so that others will join. You are all welcome. Good evening. How are you? So I think if you could remember yesterday, we have actually started discussing on some of the topic in. We started the discussion on the DNA, and I started by asking that is the DNA the same across all species of living organism? So I think that is where we started. So, and I told you that we are seriously okay. So now, I think I will now start by asking what is nucleotides? That is where we are going to start. What is nucleotides? What is nucleotides? What is a nucleotides? What is nucleotides? Please, are you with me? I said, what is nucleotides? What is a nucleotide? Excellent. They are the building blocks of nucleic acids. So nucleotides are the basic building blocks of nucleic acid like DNA and RNA. Yes, you are correct. So now, what are the components of nucleotides? What are the components of nucleotides? What and what constitutes to generate nucleotides? What are the components of nucleotides? Base and sugar? No. No. Okay. Phosphate, sugar, and base. Excellent. Suliet. Baba Eunice also said phosphate, sugar, and base. Yes, you are correct. So the phosphate, the sugar, and nitrogenous bases are the component of nucleotides. So please mention the 5 nucleotides. Mention the 5 nucleotides. What are the 5 nucleotides? What are the 5 nucleotides? As good evening. What are the 5 nucleotides? What are the 5 nucleotides? Adenine, guanine, cytosine, thymine, and uracil. Excellent. So these are the 5 nucleotides that are found in the DNA. So now the question is, which of the nucleotides are found in the DNA? Because the nitrogenous bases are 5 and we have 4 that are found in the DNA. We have 4 that are found in the DNA. So which one are found in the DNA? Which ones are found in the DNA? Adenine, guanine, cytosine, thymine, and uracil. Now this one, you are just mentioning the 5. I'm talking about what are the I'm talking about what are the nucleotides that are found in the DNA. Okay, the one that are found in the DNA, we have thymine, adenine, guanine, and cytosine. Yes, excellent. So these are the 4 nucleotides that are found in the DNA. Excellent. So now, what is Tagab's rule? 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 So please, did you set no? Chagab's rule. Chagab's rule. What is Chagab's rule? What is Chagab's rule? Okay, Chagab's rule often taught using the acronym. Okay, A to T, D to C. Explain how nitrogenous bases pair in DNA. Excellent, excellent, Edwin. Okay, is it Edmund? Excellent. Chagab's rule is a rule that trying to show how the nitrogenous bases pair with one another. So nitrogenous bases generally pair with one another. So how those nitrogenous pairs with one another is through the Chagab's rule. So Chagab's rule is a rule that trying to show how these nitrogenous bases pair with one another. So adenine pair with thymine and cytosine pair with guanine. So how many hydrogen bonds? How many hydrogen bonds are found between adenine and thymine? How many hydrogen bonds exist between adenine and thymine? That is the question. Excellent. The question is, how many hydrogen bonds are found between guanine and cytosine? How many hydrogen bonds are found between guanine and cytosine? That is the question. How many hydrogen bonds are found between guanine and cytosine? That is the question. I hope you are hearing me now. How many hydrogen bond? Four, sir? No, no, no. It's not for hydrogen bonding. Yes, the answer is 3 hydrogen bonding. There are 3 hydrogen bonding between guanine and cytosine. 3 hydrogen bond between guanine and cytosine. So, but now the question is, I want to ask this question. Is this Chagap's rule apply in DNA molecule? Does it apply in DNA molecule? Is Chagap's rule applied in RNA molecules? Is Chagap's rule apply in RNA molecule? That is the question. Is Chagap's rule apply to RNA molecule? Okay, you are saying no. Why? Okay, no Chagap's rule, or Chagap's rule do not apply in RNA molecules. That is what you are saying. Okay, so why do you think the Chagap's rule doesn't apply in in RNA molecule? Why? No, Anthony, your answer is not correct. Why do you think the Chagap's rule doesn't apply in RNA molecule? Why do you think it doesn't apply in RNA molecule? Excellent, excellent, Lecitia, because RNA is single-stranded, so therefore the Chagap's rule cannot apply in RNA molecules. So Chagap's rule cannot apply in RNA molecules because it is single-stranded. Excellent. So now, between DNA and RNA, which one do you think is more stable? Between DNA and RNA, which one do you think is more stable? Which one do you think is more stable between DNA and RNA? Between DNA and RNA, which one do you think is more stable? Okay, excellent. The answer is DNA. DNA is more stable than RNA because it is double-stranded. Okay. So now let me ask you another question, ladies and gentlemen. What is the importance of DNA? Do you know the application of DNA in how you apply the knowledge or how you use DNA in forensic science? Do you know the reason why you can use DNA in forensic science? That is also another question. Do you know how long or how DNA is used in forensic science? Before you even start thinking about this forensic science or the application of DNA in forensic science, I would now like to ask you this question. What is forensic? 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 Forensic refers to the use of scientific method and techniques to investigate and solve crimes, especially in legal or court setting. Excellent, 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 Jen. So that is actually the importance. That is forensic. So a forensic is a process that involves the use of scientific methods or techniques to investigate or to solve crimes, especially in a legal or a court setting. So in a forensic science is when you use a scientific method or techniques to investigate or to solve a crime in a legal or court setting. Because the reason why DNA can be used in forensic sciences is because it is stable. It is stable. It can take decades. It can take thousands of years. Like for example, let's say you have a group of people that survived, let's say in Zaria in the last 500,000 years ago. You can trace them, you can study them, you can be able to predict their behavior based on their DNA. So you can go there, or you can go the environment where those people survived. You can get the soil from there, and of course from the soil you can get the remnant of the DNA, and you can isolate the DNA. And once you isolate the DNA, like for example now, if you want to know me, so please get my DNA, you will know me. You know the kind of food that I'm taking, my behavior and everything. With my DNA you can get this information. So that is why the importance of DNA in forensic science, or DNA helped in solving a lot of criminal cases. Determination of paternity. Like for example, let's say now somebody just backed into somebody's house. Maybe he raped a lady or something like that. You can get the sperm. Even from the sperm of the lady, you can be able to isolate the DNA of that particular individual. And of course, usually in DNA fingerprinting, you need to have a suspect. Let's say, assume, okay, you know that between 8 to 10 p.m. you have some set of people that follow the same area, and after, let's say, some minutes, then there was an investigation as a result of maybe raping. So you can actually decide to get all those people that actually pass through that environment, get them, go to the lady that was raped. You can get her sperm. From her sperm, you can get the DNA from the sperm of the other individual, of the person that do the raping. And you can go to the lab,
get the DNA of the suspects, get the DNA from the sperm, run them on gel electrophoresis. So if the person is among those suspects, it will definitely going to give the same pattern. And from there, you'll be able to identify the individual. And also at the same time, assuming now a baby was found and we don't know the mother of the baby, but maybe there are like, let's say, 10 ladies passed through the same location where the baby was found. You can get the mitochondrial DNA, get the DNA from the mitochondrial DNA from the suspected mothers, run them on gel electrophoresis. It will give you the same pattern. So one thing, because of the high stability of the a DNA can be used, can be stored for 10 years, can be stored for 500 years, can be even stored for a millions of years. So forensic is used in investigating crimes and examining physical evidence. So that is actually forensic, and that is one of the application of the DNA. Please, do you understand why DNA is used in forensic? Because of it is high stability, and what makes it stable is because of it is double stranded nature. Please, do you understand? And if you want to know who I am, you can get my DNA, and from there you can study me, and you will know a lot from me. So please, is that clear, ladies and gentlemen? Do you understand? Please, do you understand before you proceed? Okay. So, RNA is single-stranded and DNA is double-stranded. And Chagap's rule. So, and I want to say something very important. Yeah, I think I have to say it because I saw similar questions asked in some exams, in some past question paper. Um, in the DNA, we have what you call GC content. So whenever you have a length of the nucleotide, you can be able to know the GC content. The higher the GC content, the higher the stability of the DNA. So that is why we have what we call a melting temperature. A melting temperature is a temperature that can denature the molecule of the DNA. We have, remember we said we have double-stranded DNA. So DNA is double-stranded like this. So we have a melting temperature. A melting temperature is a temperature in which the double-stranded DNA can be opened up. It will be denatured. So that's what you call melting temperature. And the higher the GC content, the higher the melting temperature. What is GC content? G means guanine and C means cytosine. So the higher the GC content, why is it that the higher the GC content, the higher the stability of the DNA? Is because, remember, between G and C it is 3 hydrogen bonding. So the higher the hydrogen bonding, then the stronger the molecule it is and the higher the stability. Do you also get this point? Do you get this point? Melting temperature, GC content, and then stability of the DNA, they are related. Melting temperature, it is a temperature that denatures the double-stranded DNA into single-stranded. So it opens off the DNA molecules from it is double-stranded into single-stranded. That is what we call melting temperature. And we also have GC content. So usually the higher the GC content, the higher the stability of the DNA. Why is it that the higher the GC content, the higher the stability? Is because the G and C fare through 3 hydrogen bonding. So the higher the hydrogen bonding, the more the stronger the molecules is. Please, is that clear? Yes, the GC bond is triple. Yeah, it's triple. And the higher it is, the higher the stability of a molecule. Please, do you understand this? Okay. So then the next thing, ladies and gentlemen, please, do you have any question regarding the DNA and RNA? I know that I sent you the video yesterday. So if you have any question, please ask. Ask me any question you have regarding the DNA and RNA before we move to carbohydrates. If you have any question regarding the DNA and RNA, that is nucleic acid, please ask. I send you 2 videos on nucleotides and the DNA molecule. So please, do you have any question? So if you have any question, please ask on the comment section so that at least we'll address it. So please explain the melting temperature again. A melting temperature is a temperature. A melting temperature is a temperature that denature the DNA molecules from double-stranded into single-stranded. I saw questions that were asked last year related to this melting temperature. So it is a temperature that often of the double-stranded DNA into single-stranded, and the higher the GC content, the higher the melting temperature and the more the stability or the more stable the DNA is. Please, do you have any question again? Please, do you have any question again? Which part? Umar Belia. Which part? Okay, sir, based on the stability, if the DNA is not stable, will it affect the outcome? Of course, if the DNA is not stable, it means there is a problem. So please, is there any other question from the DNA and RNA, that is nucleic acid? You can actually go and watch the video very well. Watch the video very well. It is all, they are all there in the videos. The Chagab's rule is a rule that trying to show how nucleotides fare with one another. Adenine fare with thymine, guanine fare with cytosine. So that is Chagab's rule. And the higher the GC content in the sequence of the nucleotides, the higher the stability of the DNA and the higher the melting temperature. Please, do you have any question? Do you have any question? No, I have said this melting temperature severally. Okay, let me repeat myself. A melting temperature is a temperature that's trying to, or is a temperature that you use in breaking down the double-stranded DNA into single-stranded. And the higher you have the GC content, it means that the higher the hydrogen bonding, because between G and C, there are 3 hydrogen bonding. So that is why before you break that bond between guanine and cytosine, you need to apply high temperature. So that is why the higher the GC content, the higher the melting temperature and the more stable the DNA is. I think I will just send you, I have a video on that melting temperature. I will send you after this class. I will send you, because I know that they have actually asked some question regarding this melting temperature. So I will send you the video. Yeah, I will send you the video so that you can watch. Yeah. So please, any other question regarding that? Do we have any question? Yes, I will send it. So please, any other question? So the next one, please, let me say this. I have seen a lot of question paper asking about the synthetic nucleotide, and I will say it. There are a lot of questions that I think, a lot of past questions that I saw, especially from DLC, that they used to ask this question. Even last year, they asked this question. And for so many years, they have been asking similar question. What is synthetic nucleotides? Even from the word when we said synthetic, we are talking about nucleotides. So now, synthetic nucleotides. What is synthetic nucleotide? The word synthetic means artificial. It means they are nucleotides that are synthesized artificially. So we can go to the lab and synthesize some of these nucleotides artificially. So that is why they are called synthetic nucleotides. Do you understand? So synthetic nucleotides are artificially created versions, of the natural building blocks of DNA and RNA. Are artificially created versions of the natural building blocks of DNA and RNA. That is what you call synthetic nucleotide. Do you understand? Synthetic nucleotides are artificially created version, artificially created version of natural building blocks of DNA and RNA. That is what we call synthetic nucleotides. And what are the importance of these synthetic nucleotides? What are the importance of these synthetic nucleotides? Number one, let's write it. Synthetic nucleotides are artificially created version of natural building blocks of DNA and RNA. That is what we call synthetic nucleotides. And then what are the importance of these synthetic nucleotides? Number one, they are used to investigate the DNA polymerase activity. Write it down. They are used to investigate the DNA polymerase activity. I hope you jot it down. What is polymerase? What is polymerase enzyme? A polymerase is an enzyme. So we use synthetic nucleotide to investigate the activity of polymerase enzyme. So what are polymerase enzyme? An enzyme polymerase is an enzyme that used in the synthesis of DNA. Remember, ladies and gentlemen, we have a nucleotides that form a DNA. So how one nucleotide joined with another? So a nucleotide joined with another nucleotide using these enzymes. So one nucleotide is added to a DNA molecules or to a growing DNA molecules using using the DNA polymerase. So a DNA polymerase is an enzyme that use in the synthesis of DNA molecules by adding the nucleotides. So we use synthetic nucleotides to investigate the activity of DNA polymerase enzyme. So that is one of the functions of artificial or synthetic nucleotides. Then another importance of synthetic nucleotides, they are used to modify the genetic material. So we have genetics engineering. Genetics engineering is one of the engineering that involve the manipulation of the DNA. So you can use synthetic nucleotides to modify the to modify the genetic material. That is to modify the DNA. Of synthetic nucleotide. One, to investigate the polymerase activity, and then second, to modify the genetic materials, and then number three, to develop new therapeutic strategies. To develop new therapeutics strategy. So these are the three importance of synthetic nucleotides. So you can use these synthetic nucleotides for development of new therapeutic strategies, like development of drugs, to develop, like, let's say, antiviral or antiretroviral drugs. You can use it. You can use it in CRISPR-Cas9, that is the gene editing technique. Gene editing techniques. What is gene editing techniques? That is CRISPR technology. Remember, there is sickle cell anemia. So with CRISPR technology, you can manipulate you can edit DNA. You can edit DNA. So if you have someone suffering from sickle cell anemia because the disease is genetics, you can just remove the nucleotide that is defective and replace it with another one. So that is in CRISPR technology. The third one is to develop new therapeutic strategies. When we say therapeutics, ladies and gentlemen, in medicine, when we say what and what comprises therapeutics? Because with the synthetic nucleotide, you can develop new therapeutic strategies. When we say therapeutics, what does that mean, please? When we say therapeutics, CRISPR technology is a gene editing technique. Most of those genetic diseases with CRISPR technology, you can go down to the gene that is having the problem or that is associated with a particular disease, and you can edit it. You can remove the defective nucleotide and replace it with another one so that the individual will become normal. So in the developed countries where they are fully developed, they use the CRISPR technology to cure most of the people that are having genetic disorders like sickle cell anemia, hemophilia, baldness, and so many others. Okay, and therapeutics, you mean treatment? Okay, so medical intervention. So like what and what? Can we say drugs? Can we say vaccine? Can we say diagnostics? All can we consider all this as therapeutics? One, the diagnostics, drugs, and vaccine. Can we consider all these three to fall under therapeutic strategies? So diagnostic, drugs, treatments, vaccines, they are all under therapeutics. So this synthetic nucleotide, they are also very important in doing that. So ladies and gentlemen, please, is there any question? I think we have shouted here, but I know that we are coming, a time will come where we'll be start solving past question paper or biochemistry and other courses that we are doing tutorial for some of you that are members to my YouTube channel. Remember, we told you that for those of you that are interested in our courses like human physiology 225, human physiology 227, biostatistics, and anatomy, if you are interested in these courses, so I advise you to try to watch my to register with my YouTube channel so that at least you can have access to all these videos. But for biochemistry, it is free. You don't need to make any payment to watch the videos, but for the other courses you have to pay if you want to watch the video. So if you are not a member, I advise you to try to register with the YouTube membership. And very importantly, we have started solving past question paper, and already there are some solve past question paper in the YouTube, especially for those other courses, even this biochemistry. But we will I will release all those questions near exam time, because this is the best time to start reading. Just read, focus on your notes, focus on the videos, focus on the materials to have the general knowledge. So try to register with the YouTube membership so that at least you can have access to other videos. So thank you very much. I'm going to stop from here, and please, since you have no questions, then we can stop here. Prosper, I saw you have been trying, so God bless you. So thank you very much, ladies and gentlemen. See you next. So we are going to continue with carbohydrate next time, and I already sent you the videos on carbohydrate. Please try to watch. Thank you very much, and bye bye.